Yeah, because you're fast, you could have been a sweater. Oh, how was that? How was your going?
I don't know. Let me see the case. Is that a new shirt? Is that a new shirt? I like How do you zoom? How do you zoom?
It's almost 10 after. We might as well begin. All right? Hello, Aaron, David, Benjamin, and Laura, my sweethearts, and my grandchildren. I am your professor, and I'm ready to begin this lecture. Now, this is my last lecture, and I can talk in my normal accent. I do not have to try so hard to be understandable. I can say, I can park my car in Harvard Square if I want to. And I can tell my dog to stop barking. Freedom! You ready? Therese asked me to remind you that you should bring a pencil and eraser to the final exam, because last time apparently she had to leave the room to go get them, okay? I made some of that up, but at any rate, last time I explained the individual steps that occur in the lymph nodes and where the antibodies are made. Most of your antibodies are made in lymph nodes. I didn't want you to think that's your only lymphoid tissue. It's not, but it's the major one. So most of the antibodies are made in the lymph nodes. And when I talked about that, I talked about the role of the APC, which is the antigen-presenting cell, the Th cell, which is the T helper cell that's activated and clonally expanded by the APC, the B cell, which when it binds to the Th, it becomes clonally expanded and it makes antibody. That means that the B cell becomes the plasma cell that makes antibody, and I told you about that. I also told you that the antigen-presenting cell, in order to present the antigen, it has to present it on a protein on its surface called MHC2. So MHC2s are found on the surface of all APCs, B cells, dendritic cells, macrophages. Okay? Then I told you that in order for there to be a tight binding between the TH cell and the APC, doesn't make any difference whether that APC is a dendritic cell, a B cell, or a macrophage. In order for that TH cell to bind tightly, that TH cell must have on its surface, and it always does, a protein called CD4 that binds to the MHC2. Otherwise, the TH cell will not be optimally activated and clonally expanded. And then I told you that a certain virus called HIV binds to the CD4 and as a consequence can enter the TH cell and destroy it. And that is why HIV causes inhibits the immune response. It's because what it really does initially is it causes the death of TH cells. And the TH cells are required for the entire immune response, not only making antibodies, but also activating the cell-mediated response that I'll talk to you about today. So if I kill those TH cells, I no longer have an immune response. Okay? And I told you that. I also told you in that humoral response, I told you that when antibodies are made, they're made in 2 stages. I talked to you about the primary response. That's the first response. And then at the second introduction of the antigen, for example, a booster shot, okay, you get the secondary response. It's the secondary response that's really the protective one, because you get a huge increase in antibody, and it's IgG. That first antibody is mostly IgM. And the reason why you get such a rapid and huge increase in antibody in the secondary response, I told you, is because what you have there as a consequence of the primary stimulation of B cells, what have you accumulated? What kind of cells? Memory cells. And they're the ones that will go to the plasma cells and be activated. They're responsible for it. Okay? Then I described the steps that occur during immediate hypersensitivity reactions. These are the allergic responses that you're used to. Within minutes, you respond. Minutes. Okay? It could be to a bee sting. It could be to penicillin. It could be to food. What else? One more thing, like that I mentioned. Pollen. Pollen too. You start sneezing or your eyes will water. This all has to do with an immediate type hypersensitivity response, and that's mediated by IgE. And I told you what happens. I told you that the allergen, which is the antigen that induces the hypersensitivity response, I told you that the allergen activates the APC cell so that it can activate the TH cell, so that it can activate the B cell, so that it can become a plasma cell. But guess what? The allergen doesn't induce IgG. Unfortunately, the allergen induces IgE. Okay? And right away, you have to know that I didn't tell you the whole story. I said TH cell as if there's only one kind. That's not true. There's a whole spectrum of TH cells, and some of them produce cytokines, this is an aside, that activate IgE. Okay? So you have this IgE that courses through the bloodstream, as I told you, and finds these mast cells. And I told you that the mast cells was in connective tissue, lining the blood vessels, lining the bronchi, in your eyes in the connective tissue, in your nose in the connective tissue. Mast cells are everywhere. The IgE, I told you, binds to mast cells because mast cells have a receptor for it, I told you. And then I told you when the IgE binds to the mast cell is sensitized. A sensitized mast cell has IgE bound to it. then the next time that allergen comes in, I told you, it binds to the IgE that's on the mast cell and triggers the release of what?
Histamine.
Histamine and leukotrienes. That's where I'm about to stop today. Okay? Today I'm going to finish that story, and it will be very quick. I'm going to tell you about the cell-mediated immune response and the DTH response, vaccinations, and if there is time, I'll tell you about inflammation. If it's not, I won't. Correct? There you go. And the review sessions next week are here, and you're all invited to come. Okay? You have any questions about the last lecture? Okay. Okay. Here's where we left off on the last lecture. I have now mast cells. Actually, I got a special pen for this purpose. I thought it might be better. We'll see. This is the mast cell. It's possible that it's by writing and not the pen. I don't know. But I got a pen that might improve the writing. That doesn't make sense, does it? And this mast cell is sensitized. What does that mean? It means it has IgE on it. Okay? And the reason it does is because the mast cell has a receptor for the IgE and it binds to it. And then when that allergen is produced again, it could be in the food, it could be something I breathed in. See, when the allergen is introduced, or could be in the bee sting, when the allergen is introduced again, it binds. There's the allergen. This is where I drew last time for you. It binds to the IgE. That sends a signal into the mast cell. And as a consequence, as I told you last time, the mast cell will secrete histamine. It will secrete histamine and compounds called leukotrienes. Okay? Now, I do not have the time really to go into the details of how they work. So I want you to refer to these as inflammatory substances. They can be called inflammatory substances. Sometimes they're also called vasoactive substances. And I thought you should at least know those two words. So the histamines and the leukotrienes, you can refer to them as either inflammatory substances or vasoactive substances. What do they do? I'm going to tell you, although there are some differences between them, I cannot emphasize those points in this lecture. So I just want you to consider what they do as a group. Okay? What they do is they cause dilation. What does that word mean, dilation? Expansion. Expansion. They cause dilation and leakage of blood vessels. If your blood vessels dilate and leak, if they leak, will I get any swelling?
Yes.
Correct. So if I have my blood, it's when I taught you about making lymph. If I have too much leakage, I get a lot of fluid coming out in the interstitial fluids. And these substances can cause that. And as a consequence, you get swelling. So you might get swelling as a consequence of those two. Okay? In addition to that, the dilation allows more blood to come to that area. Is that correct? Mm-hmm. More blood can come. If more blood comes to the area, will that change the color?
Yes.
Absolutely. What color will it become?
Red.
Red. So you get the redness as well because of that. In addition to and how about the nose? Your nose might drip too because of all that fluid coming out. Okay? In addition to that, it depends where the allergen goes, right? If it goes into your nose, it will cause that. Okay? In addition to that, that allergen will increase, it will increase mucus secretion. Okay? What kind of epithelium secretes mucus? The mucosal epithelium. So it stimulates the mucosal epithelium to secrete mucus. That's one of the reasons that, who coughed just now? Who coughed? Raise your hand. The reason you coughed is because mucus was going into your throat, into the airways, and you had to cough it out. So you will cough too as a consequence because of all that mucus. It's going to go someplace, and then you might spit it out. Okay? In addition to that, those vasoactive or inflammatory substances, they also, if this occurs, if those mast cells that are activated are along the bronchi and not in your nose or your eyes, depending upon where the allergen goes, then these two substances, histamines and leukotrienes, can cause contraction of the smooth muscles in the bronchi. Let me ask you something. If the smooth muscles in the bronchi contract, what will that do to the airways? Will it constrict them or dilate them? Will constrict them, and you find it trouble in breathing. And this is what happens when some people suffer from allergic asthma. Okay? So, that's the immediate type hypersensitivity response. Does anyone have any questions? In the book, I tell you how to treat it, but I do not have the time in this last lecture to go into that. So if you want to know about treatments, it is described in the book. Do you have any questions about that before I go on now to the cell-mediated response? Because I also want to talk about vaccinations. Okay? How many of you have pollen allergies? It's not uncommon, is it? Okay. It's a pain in the neck. No, it's a pain in the lungs. All right. Now I'm going to talk to you about the cell-mediated response. The cell-mediated response. The cell-mediated response begins when cytokines, remember what cytokines are? What are cytokines? They are the signaling molecules that are secreted by the TH cells and other cells. But I'm concentrating on the TH cells, okay? When these TH cells will secrete many different kinds of cytokines, 12, 13 different kinds. Hi. How you doing, James? Good. Many different kinds of cytokines, okay? And when they're secreted by the TH cells. These are the ones that I am interested in talking to you about. Those TH cells must be activated and clonally expanded. Okay? What does that? What clonally expands and activates the TH cells? APC cells. This process is beginning exactly the same way the humoral response began. No difference. But the cytokines are going to be different, because I told you there are different kinds of TH cells. And these cytokines, what they're going to do is they are going to clonally expand and activate as well lymphocytes called TC cells. This is the other part of the immune response. Some of those TH cells are going to go over to the B cells and say make antibody, but some of them are going to secrete a lot of cytokines that will activate and clonally expand TC cells. Okay? In addition to that, these cytokines, some of them will activate, and I'll describe this to you briefly, activate macrophages. And when those macrophages become activated by the cytokines, they are better killers. And I am going to explain both of that to you now. Okay? I'm going to start, maybe that's not a good pen. I'm going to start with the TC cells. That's not, Back to the old pen. I'm going to start with the TC cells. I have clonally expanded and activated them with the cytokines. What do they do? Okay? They are going to do two things that I'll describe to you. One, these clonally expanded activated TC cells, they're going to kill virus-infected cells. You got to do that because the antibody cannot get into a virus-infected cell to kill the virus. The antibody can only inactivate free virus. In order to mount an immune response that's active against virus-infected cell, you need the cell-mediated response. One of the problems in developing developing a vaccine for the HIV virus is you've got to have a vaccine that will stimulate not simply the humoral response, but also this response, or else you will not be able to develop an adequate vaccine for the HIV virus, because it lives inside, it replicates inside the TH cells, okay? So if you want to get that, you also have to be able to activate the cell-mediated response. In addition to killing virus-infected cells, the TC cells that are activated and clonally expanded, they will kill foreign tissue. Now, the foreign tissue is a graft. The reason why you reject grafts, foreign grafts, is not because of antibodies. That's not nearly the problem. The real problem is your TC cells, which will bind to and kill those foreign cells, okay? And I will now try to explain to you how that occurs. For that, you need to turn your page to a fresh page. Okay? Now, I'm going to draw for you a cell, part of a cell. And on this cell, I'm going to draw for you a receptor. This receptor is similar in some ways to the MHC2s, but it's not MHC2. This receptor is MHC1. MHC1 is also a protein. MHC2s are found in what kind of cells? All APCs. No exception. But MHC1s, on the other hand, they're found everywhere. They're found on all nucleated cells in your body, even the lymphocytes. So the MHC1s are found in all your nucleated cells. And if you were to examine those MHC1s, you would see that there is bound to it a peptide antigen. These peptide antigens there are no MHC1s that do not have a peptide antigen bound to them. So they have a peptide antigen bound to them. Okay? This peptide antigen, let me tell you what it is. It is a sampling. It's a random sampling of the proteins that the cell makes. In other words, all of your nucleated cells have on their surface a random sampling of peptides saying, this is me, I belong to you. Okay? These are your peptides, don't touch me. So the immune system will not touch those cells if they have your peptides and if they have your MHC1s. Okay? Now, when a virus infects a cell, that cell is making viral proteins because it's making new viruses. And that cell will then put on its surface a sampling of all the proteins it makes, including the viral proteins. So a virus-infected cell differs from your other cells in that on that MHC1, you'll find viral proteins as well. Okay? All right. Now, here we have a TC cell. Okay? It's going to be looking around for a foreign protein. If it's a virus-infected cell, it'll be looking around for the viral antigen. If it's a transplant cell, then it will be looking around for a foreign MHC1, and possibly a foreign protein. Okay? All right. So let's assume it's a virus-infected cell. That TCC cell has, I thought it was a TC steel. That TC cell has on its surface a T cell receptor, just like the T8 cells do, a TCR. Okay? Some of those TC cells have a T cell receptor that recognizes that viral antigen. Okay? The receptor also is a foreign antigen. It also recognizes and binds to the MHC1. Okay? This is quite similar to what a T8 cell does. The T8 cell will bind to a foreign antigen on an MHC2. But the TC cell binds to a foreign antigen if it's a virus-infected cell, or the MHC1. Okay? Now let me ask you something. Do you remember that when the T8 cell bound to an antigen-presenting cell, the TCR was not sufficient? What else did it need? CD4. Okay? These cells have something similar to that. The TC cells have a protein on the surface, but it's not CD4. These have a protein called CD8. And because of that, the TC cells are called CD8 cells. What are the T8 cells called? CD4 cells. So these are CD8 cells. And the CD8 does what the CD4 does, but it binds to MHC1. Not 2, but 1. Makes that strong binding. Okay? Where do the cytokines come into all of this? Okay? You have cytokines that were produced by the T8 cells when the immune response was begun. These cytokines are doing two different things. One of the things that those cytokines from the T8 cells are doing was to clonally expand the TC cells that are bound to this antigen. Those TC cells can only be clonally expanded when they're bound. They can only respond to the cytokines when they're bound. Therefore, the only TC cells that can be expanded are the ones that have a TCR receptor that's bound. And you can clonally expand and get many, many of these TC cells now. In addition, the cytokines do a second thing. In addition to clonally expanding the TC cells that are bound and therefore recognize that viral antigen, in addition to that, what the cytokines do is activate the TC cells to secrete toxins. Now, the TC cells, by the way, will secrete those toxins even if they're not activated, but you get much more secreted when it's activated. And so I am going to come out of this cell with a toxin. That toxin will not diffuse through and hit other viral infected cells. These cells are right up close to each other. That toxin is going to impact only on this cell. Only on the cell to which the TC cell is bound. As a consequence, it will kill the virus-infected cell or foreign tissue. The foreign tissue has an MHC1 that's different from yours, and that TCR recognizes the difference. So you reject your foreign tissue for two reasons. One, it may have a foreign antigen, but two, the TCR becomes activated when it hits a foreign MHC1. That's why your tissues have to be, the transplant has to be matched. What they match is the MHC1 and MHC2. Yeah, Ben. Did you have a question? Okay, Ben, after class ask me, okay? So that's the TC's part, okay? But the cell-mediated response also, in addition to activating the TC cells, the cell-mediated response also activates macrophages. In other words, what activates the what do the T8 cells produce that activate macrophages? Cytokines. So some of those cytokines, they're not all the same, obviously. Some of them are going to clonally expand and activate the TC cells. Other cytokines produced by the T8 cells will activate the macrophages. Okay? When I say activate the macrophages, I mean make them better killers. The macrophages must first phagocytize the pathogen, but then it helps if they're activated to kill. Okay? What activates macrophages to phagocytize pathogens? Opsonization, correct? So the antibody and the activated complement will coat the pathogen. The macrophage will phagocytize the pathogen. You remember that? But the killing is activated by the cytokines produced. They'll kill, but you get a tremendous activation by the cytokines. So it activates it, okay? So you must think of, this will be your almost last drawing. You draw what I tell you to draw. Because macrophages are quiet unless they're activated, okay? So here is a quiet macrophage, okay? So you have those, your macrophages aren't always active. They're activated in part by the immune response, I'm going to tell you that now, but also during phagocytosis, that activates them as well, okay? But the immune response makes them even more active, okay? So I am going to take some cytokines and activate the macrophages. Who produces the cytokines? The T8 cells as part of the cell-mediated response, okay? And that macrophage is just tremendously activated. It can also be activated without cytokines when it phagocytizes, because you always have macrophages that are protecting you, but there's an increased killing activity due to the cytokines. As a consequence, this macrophage wakes up and becomes, those are teeth. Somebody wants me to raise this up. That macrophage is now a killer. There's a lot. Okay, there is a lot. No, now you remember the question, Ben. What? It looks like another face? Leatherface. I don't know what Leatherface is. Okay. There is a lot known about what macrophages are activated to do when they kill. I do not have the time to tell you this. It's not a mystery, okay? The macrophages are activated to kill. So you can think of this macrophage as sleeping, and you can think of this macrophage as ready to kill, okay? Now, when the macrophage is stimulated to that extent, not only will it kill the pathogens better that it has taken in, but in addition to that, drooling out of its mouth are toxins. How about toxic, why don't you write, not toxin, toxic substances. So activated macrophages produce toxic substances that come out when they're fully activated. And the cell-mediated response can cause this to happen. What happens to these toxic substances is that they injure the tissues in the area. And when they injure the tissue, and they can destroy the tissues too. They can also destroy the tissues. So you do not want too many of these activated macrophages. They can harm you if there are too many. It has to be tightly regulated. And not only do they injure and destroy the tissue, but they will cause an inflammation. So, these activated macrophages that secrete the toxic substances cause an inflammation, destroy the tissue, okay? That response has a name. What's it called? The DTH response, absolutely. It's the delayed type hypersensitivity response. It's due to these activated macrophages. It doesn't happen all the time. It only happens when there's an overproduction of these toxic substances. It is a possible bad outcome, but not always the case. And I want to give you a short list that you will be familiar with of symptoms due to those activated macrophages destroying local tissue and causing an inflammation. Okay? One is poison ivy. 2 days maybe after you've been exposed, something like that, maybe 3 days, 1 to 3 days after you've been exposed, you get an inflammation, you get a reddening, you get an itching. That's due to the activated macrophages causing that inflammation because they're destroying or hurting the tissue. If I have a chance, I'll tell you why it becomes red, inflamed, and itchy. I don't know if we'll have a chance, but if we do, I will. Okay? In addition to that, when you get to be my age, you're going to get, if you do not, it's not simply brushing. Brushing won't make it. If you do not stimulate, use stimulants between your gums, okay, you might develop an infection of your gums that can get so bad, it will be called periodontitis. And when you get periodontitis, you destroy the connective tissue in the gums holding the teeth in. That's why people lose their teeth. They don't lose it so much anymore. Okay? I've been going to the dentist for years for periodontitis because when I grew up, no one told me to floss or to use those little stimulants. I just thought, well, you brush your teeth, that'll be good enough, but it won't be. Okay? Now, what's the periodontitis due to? It is a DTH response. You've got a cell-mediated response in your gums that's due to an infection. So there are bacteria that infect the gums, and as a consequence, there's a cell-mediated response. There's an overproduction of these macrophages. They're secreting, in this case, an enzyme that destroys the connective tissue, and you've got periodontitis. How many of you intend to go to dental school? Then for those of you who intend to go to dental school, I will tell you that the name of that bacterium is Bacteroides. Those of you who do not, that's one of the names, there are two. Those of you who do not go to dental school, you will not be asked on the exam what the name of that bacterium is. Okay? Now, there are other diseases whose symptoms are due entirely to a DTH response. Okay? I just want to mention them. One is tuberculosis. One is tuberculosis. You will learn in some other course what tuberculosis is. It's a destruction of lung tissue, essentially, and that is due to those activated macrophages. Okay? Not to the bacterium that causes tuberculosis. That's an innocent bystander. It's due to the DTH response. Okay? Also, leprosy. At some other time, I told you this before, you will learn about leprosy. The symptoms are due to a DTH response. Also, the tertiary stage of syphilis. You have destruction of neural tissue due to a DTH response. Also, and I thought I would mention this because it's significant in Indiana, a disease called histoplasmosis. Now, histoplasmosis is a yeast infection of the lungs, and you get it by inhaling bird droppings. Well, you do, because the yeast are in the bird droppings. So people who live out in the country on farms might develop. I had a good friend actually who got histoplasmosis, and it's a yeast infection of the lungs, and it's due really to a DTH response. If the yeast do not make a toxin that kills lung tissue, it's due to an overexpression of the cell-mediated response. Okay? We're doing fine. Now, that's all I was going to tell you about the cell-mediated response. Do you have any questions you want to ask me before I go on to vaccinations? Yes?
I just wanted to clear two things. You said that TH cells...
Listen to his question. It may be on the exam.
TH cells will produce cytokines which activate the TC cells.
Correct.
And TC cells produce cytokines to activate macrophages?
No. The cytokines that activate the macrophages are produced by the TH cells. The TH cells are the lymphocyte that activates the humoral response and the cell-mediated response. They are central. The TC cells are activated by the cytokines produced by the TH cells. Okay? And the macrophages are activated by the cytokines produced by the TH cells. Okay? Yes, Ben? What? I'm sorry, you have to say it loud enough so I can hear.
When the cells that have virus are killed,
Yeah, what happens to them?
no, like how it kills the virus, it's kills the virus, destroy them.
Ben, for that you have to know something about viral replication. Those cells are not storing viruses. Those cells are making viruses, and that's why some of those antigen are on the surface. And as a consequence of making virus, the cell will be destroyed, the virus will be released and infect other cells. So what the TC cell does is kill the virus-producing cells. If any virus is released, it will infect the cell that will be killed, but eventually the virus will be killed. If it's effective. If it's effective. Yes? Any other questions? Now, all right, I know you want to go home, but it's just a few more minutes, okay? And if you're good, I may let you out early, in which case I will have retired early. So I'm going to tell you something about vaccinations, okay? Vaccination. All right. There are two types of vaccinations. And I think you should know this because not only will you going to be vaccinated yourself, some of you will raise families, and your children will have to be vaccinated. And in this country even, not enough people appreciate the importance of vaccination. Okay? And if you read the newspapers, you will read that in the cities like in Detroit and some others, shouldn't mention a particular city, in various cities, the children aren't sufficiently vaccinated because the parents really do not understand the importance of vaccination. So I want to tell you a little bit about vaccination. Okay? There are two types of vaccination. One type is called passive. So this is passive vaccination. And when someone is passively vaccinated, what they do is they receive, they receive antibody. In other words, it might be gamma globulin. Gamma, I don't know if I told you this, but gamma globulin is another way of saying immune globulin, immunoglobulin. So gamma globulin is the same thing as IG, okay? Passive in vaccination, you receive serum, globulin, that was made in either another person or another animal, okay? For example, in the horse. But now they like to do it, they like to take it from people, more so, because of a disease you will learn about later called serum sickness. You won't learn it here, okay? And you get passively vaccinated for a variety of things. Okay? What you have to remember about passive vaccination is it lasts only at the most a few months. This is not a long-term fix, but it's an immediate fix. You're immediate immune because you get the antibody right away. So for example, some of you may become passively vaccinated against hepatitis A. Okay? Now, you may or may not know what hepatitis A is. It's a virus that's spread via fecal contamination. Okay? It's a virus that's spread by fecal contamination. You can get it in foods. So, for example, you go into a restaurant and someone has hepatitis A, is preparing your food, and they don't wash their hands, and it gets into your food, you can get hepatitis A. If you get hepatitis, what's the major symptom? Jaundice. Right? Become jaundiced. If you have a liver problem, you become jaundiced. Remember I told you about liver making the bile? If there's a problem in making the bile or putting it into the bile duct, some of that bilirubin will go into the skin, and you can get jaundiced. Okay, so that's one of the symptoms of hepatitis A. And sometimes if you're exposed to hepatitis A, you're asked whether or not you will accept some immunoglobulin, and you get it. And that will prevent the virus from growing in you. Another example of passive immunization is rabies. Let's suppose that you were bitten by a dog, and it was clear that the dog was rabid. Okay? Then you may be told, look, we may be able to immunize you permanently for a long term against rabies, but we want you, and I'll tell you about that in a moment, but we want you to be right away immune. We don't want that, it's a virus. We do not want that virus being able to get into your brain, into your neural tissue, and cause paralysis and possibly death. Okay? As a consequence, we want to give you antibody to the virus. It's passive immunization, given right away. How about tetanus? Now, this isn't always done, but if a person is clearly got a problem, that is, tetanus is caused by a bacterium that's in the soil, but if you have a rusty nail or something and you have a deep gouge or cut, and the doctor said, I know where you've been, you might have been infected with the tetanus bacterium, then you might get antibody to it. Right away, passive, okay? So there are various examples of passive immunization. Here's another one, botulin. You know, botulin is a poisoning, it's a food poisoning. It potentially can kill you. It hits the nervous system. If someone has symptoms of botulin poisoning, then the doctor will say, I will passively immunize you. Give you an antibody to the toxin so that at least you have a chance. Okay? So passive immunization is done for tetanus, for botulin, for hepatitis A, and a variety of things. But it only lasts a little bit. It's an immediate cure, but it only lasts for a while. The other kind of vaccination is called active vaccination. Now, in active vaccination, you do not give the antibody to the person. In active vaccination, you inoculate with the antigen, prepared in different ways, with the antigen. Influenza virus vaccinations are active in vaccinations, okay? And in active vaccination, you give the antigen. How will the body respond? With making antibody and possibly a lot of a cell-mediated response. Both responses occur. So now the body is making the recipient is making the antibody, and active vaccination can last for years, sometimes a lifetime. Years and sometimes a lifetime. I'm going to give you some examples of active vaccination. These would be children vaccinations. You must vaccinate the children accurately. Before I do this, I just remembered something that was interesting. One more example of passive immunization. What? Do you know what I'm going to say? One more example of passive immunization. Yeah? What? A booster shot, okay. The booster shot has to do with active, okay? There's one more example I thought would be relevant for passive immunization, breastfeeding. Passive. If you have a mother that's immune to chickenpox because she's had it, if she breastfeeds her baby, the baby is immune to chickenpox for a few months. Why? What antibody, what class of antibody comes across, is secreted into the milk?
IgA.
IgA, that is correct. And it protects the baby from certain kinds of infections, including chickenpox. Yes, Oscar?
Yeah, when you're injected with the passive antibodies, why wouldn't you have memory cells that would last more than a few months so that you'd have protection for years?
Okay, Oscar wanted to know why it lasts only for a few months when you get an injection of antibody. Antibody does not stimulate the immune response. It does not stimulate the TH cells. It doesn't do anything like that. In order to stimulate the immune response, you need antigen. Okay? All right. So, children's vaccinations, what do they include? DPT. 2 or 3 times. Okay? DPT, that stands for diphtheria, pertussis. Do you know another name for pertussis? Whooping cough. Diphtheria, pertussis, and tetanus. So all children get a DPT, more than one, because you need to get the booster shots as well. DPT vaccinations will give them protection against diphtheria, whooping cough, and tetanus. They may not last for the rest of your life. People don't understand this. Let me give you an example. You really have to be vaccinated against diphtheria and tetanus once every 10 years, or else you won't be immune. See? So a lot of people may not be immune now to diphtheria. The reason that diphtheria is not a problem is the children are being vaccinated, so that no one has diphtheria around you. But if you're still young enough, but if you weren't vaccinated, if you didn't have a revaccination for diphtheria, and you were, say, 30 years old, and the child next door had diphtheria, you'd get it too. Okay? Let me give you an example of when that happened. In the early 1990s in Russia, you may not remember this, but the Soviet Union collapsed around in the late 80s, okay? And it was no longer this one central country, okay? And when that happened, there was problems in vaccinations. There was no regulation, okay? So in the early 1990s, before that even, there was less or no vaccinations against diphtheria. In other words, the children weren't being vaccinated. As a consequence, some diphtheria. You think of diphtheria as something, who's getting in it now? Some diphtheria came up, and in the early 1990s, 50,000 people were diagnosed with diphtheria. So those it's important to keep up your vaccinations. Did you know that in this country, in the 1930s, diphtheria killed more children as a consequence of infection than any other disease? Thousands and thousands and thousands of children died because of diphtheria. So it's important to keep up with these vaccinations. In addition to diphtheria, children are vaccinated with something called MMR. This is measles, mumps, and rubella, which rubella is measles, and that's German measles. The problem is that worldwide, now you can prevent measles. The real measles is this one, rubella, that's a killer. You can prevent measles by vaccination. But worldwide, close to 2 million children die a year because of measles, because there isn't sufficient vaccination. I want you to know that I was not able to retire early. I was also going to tell you that I would answer any of your questions, even tell you what the weather was going to be. I had a lot of jokes for you, but it's a little after 11. You can come to any of the review sessions next week. And whatever, thank you. The toxin can travel throughout the body. So the tissue that's killed is not simply the tissue in the throat. You can get kidney problems, you get organ failure.